Okay, hello everyone, welcome back. So now it's my pleasure to introduce our next keynote speaker. Also in the topic of microbiome data analysis, we have here Dr. Aura Raulo. She's a microbiologist and network scientist based at the University of Oxford. And Dr. Raulo develops and applies graph-based statistical models to model microbial transmission processes in contact networks. That's all I'm going to say. I don't want to give you any spoilers. And thank you very much, Raulo.
Okay, thank you. Nice to be here. I'm going to be telling you about a biological process and how to model it. But I studied the spread of microbial communities in contact networks, but I'm actually interested in spread of complex spread of all kinds of things. So all of these methods are quite applicable for other things like spread of information, spread of values, spread of maybe resources. And yeah, so if you have ideas on how to apply these models on other kind of data, then I'd be really interested to know. But yeah, but I'm a biologist, ecologist by training, so this is like I'm very, very much coming from the background of studying microbial ecology. So many things spread from individuals to another in networks. We are quite familiar with some of these processes. There's many models to study this, like information or opinions. Here's a study on political opinions spreading in social media and this phenomenon that we can be driven into these echo chambers or polarization that our networks can actually limit the spread of diverse information by their structure. That's quite interesting and quite valuable, important thing in the world at the moment. Let's see if I can make this work. Yeah. Other things can also spread like resources like money, but here's just a case study on food sharing in a village community, so people can give each other some valuable resources. And of course money, and there's like lots of interesting network analysis studying like how money flows across kind of trading networks. But most, I would like to just, most importantly, and maybe the case that we're all most familiar with is the spread of pathogens, epidemics. When we think of networks, maybe often we first think of the spread of viruses for a very, the reason that we've all been influenced by COVID just very recently. And there's lots and lots of research on how to trace the spread of a single pathogen across a network of people or animals and people. But what people don't always think about is that when we come to contact and we share microbes with each other, bacteria, viruses, fungi, all kinds of things, that first of all, most of them are not pathogenic. Great majority of them are either neutral or actually beneficial to your health. And second of all, when we come into contact, we do spread thousands of different things. We don't just spread one thing. We can, of course, model the spread of one single virus or one single bacteria, but how do we go about modeling mixing and spreading of whole communities of interacting microbes? That's a mathematically very different process from just tracing the travel of one virus from place A to place B. And that's very valuable in this kind of emerging new study of spread of microbiomes, because we know we have these microbial communities that are very important for our health. And they do also spread in social contact, just like disease, but that's what we can't use really the disease spreading models to study this process, because this is very different spread. It's a spread of multiple things at the same time in both directions. It's like symmetrical homogenization, a bit more like mixing paints, like I have red paint, you have yellow paint, together we become orange, than it is to like passing on a baton or something like that. So how to model directional spread versus symmetrical spread. This is like a lot of my career is just based on just building models to find signals of contact-based sharing or homogenization of communities in social contact. And my study system is the mammalian gut microbiome, specifically mouse microbiome. I study this mouse system or a social system of wild mice that live wildly in a forest. I'll tell you a bit more about it in a bit. But basically, I don't have to, maybe many of you already know, but that I don't have to like give you a like whole introduction to gut microbiome. You probably read about it. It's a very important community of microbes living in our guts, in the guts of mice, in guts of every animal. And they are not just diverse ecosystem, they're also very influential for the function of the body. So they affect this like unique combination of microbes. It's like a cocktail recipe you have inside your gut, and we all have our own specific microbiomes that affects our bodies, our metabolism, immune system, even things like cold tolerance and central nervous system, and affecting our brain and our behavior. So it's really important part of our body, but we know very little about what forces actually govern this variation. Like why do I have different microbiome from yours, and so on. And that is because it's not just, we can't just explain microbiome variation with traits of the host, like genes or diet or immune system. It's because it's not just a trait of, like the microbiome doesn't just belong to the host, because it's actually constantly spreading between different hosts. We need also between individual processes to explain microbiome variation. And that means that the microbiomes are actually ecologically thinking a bit like an island ecosystem. They're like meta communities nested inside physiological systems. So often when we think of ecology and physiology, we think of physiology like bodies inside ecosystems. But this is actually ecological systems inside bodies. And we can use the social contact network of host animals or individual humans as kind of the roadmap or the bridge map of this archipelago where these ecological communities assemble. And luckily, there is a whole last century has been rich in developing ecological models on how ecosystems accumulate on fragmented landscapes like islands. So we can use that knowledge of ecological complex communities accumulating in fragmented landscapes and combine that with what we know about network spreading processes from pathogens, and try like bring those things together to develop a model where we like look at how microbiome combinations of individuals arise as a function of their position in the social network. The way we do this is we can make models where we predict microbiome composition or a similarity of microbiome, for example, between two individuals with their proximity in various contact networks. They can be overlap in space, they can be actual contact networks like social networks, or they can be ecological association networks. There's all kinds of networks that describe contacts between individuals. And I'm going to show you now a case example of studying microbiome transmission networks in wild mice. There's 3 questions I'm going to try and answer. How to find a signal of social contact-based microbiome mixing. Then if you do find one, what kind of microbes are then spread through what kind of contact networks? Can you like disentangle that? And what kind of positions in a transmission network accumulate the most diverse microbiome? And yeah, I'll get to the third one if I have time. I'm not entirely sure, but let's see. So my study system, as a biologist, I also spend a lot of time actually outside in rubber boots and stuff. So I study these cute little animals. They're called wood mice, and they live wildly in Wytham Woods Forest in Oxford, where I work. And this is just some examples of their social behavior. What do they do then together? Well, apparently they float in the air, they have a party, gravity doesn't really apply to them. But most importantly, if you look at the left side pictures, they kiss. They sniff each other's noses, they lick each other's mouths, which is behavior that all mammals do, including us. We have this weird tendency of licking each other's mouths. And we don't stop to think why we do it because it feels good. But actually, many scientists have speculated that the reason why this behavior, kissing, evolved is a lot to do with spreading bacteria, like healthy bacteria between us, and especially spreading those bacteria that are allergic to oxygen, who can't tolerate air. So it needs a really intimate contact to get from one individual to another, and there's not so much air in a kiss. So that's why we can think you need to have a little bit of kisses to spread some bacteria. The data sets I'm using here are these things I collected during my PhD and postdoc. So in these like like kind of 1.5 to 2.5 hectare areas, I trapped all the mice, or like 95% of the mice that lived there, inject the microchip under their skin and release them back to where they were from. So they just kept on living their normal life in the forest. There's no cages, nothing. They're just wild mice in a wild forest. But we can follow them with this these loggers that like are tracking devices to track their movements when they have these microchips. And then we also collect, while we trap them, we collect their poo. Um, uh, it's an easy way of getting a lot of data. That's why I love mice. So instead of studying like tigers, where you get like 10 data points, if you study mice, you get like 900 samples. And then use the feces to do 16S amplicon sequencing to profile their gut microbiome, and we use the DADA2 package to inference amplicon sequence variants, which are a little bit equivalent to species in bacteria, as you might know. And then use the OMA workflow that many of you have here discussed for data filtering, normalization, and calculation of pairwise community similarity. And just one of the earlier talks discussed about this abundance versus prevalence, or like presence absence data being just as good as abundance data. And I think we have found something similar. So when we look at this pairwise, when we calculate this pairwise community similarity metrics, we can do it in a way where we look at presence and absence, like use checkered index of how many of the same species you share, of all the species you have, or we can do that in an abundance weighted way, or in other ways weighted ways. And we have found out that you always get the clearest signals with just the presence absence one. So the abundance information, at least in this process, is just noise, which makes sense ecologically, because actually microbes, when they spread, their abundance in your gut is not really dependent on how much transmission, how much spreading is happening, but more like how well they grow there once they get there. So it's more like what you have, what species you have is dependent on transmission, and then how much you have them is dependent on other stuff that happens inside your gut later, like what you eat, for example. So then this, oh, I'm missing the logger picture here, but there should be a tracking device picture there. So also, in addition to microbiome data, we have this really cool tracking data that is basically spatiotemporal like occurrence data, who was where when, and we can use that to derive both home ranges and social networks of these mice. So we can look at like kernel distributions of each mouse, like where they are, and they are very territorial animals. They always hang out in the same, roughly 500 square meter area, so very small place, and you can always see the same mouse in the same area. They don't really go anywhere. And then you can use these home ranges to look at home range overlap. So how much two mice are exposed to the same exact space. And then you can use this data to develop a social network based on how often two individuals are actually in the same place at the same time, or within a short time window. And that's important, because in this species, social associations and spatial overlaps are actually quite weakly correlated, which is very, very important for my aims, because the fact that they hang out socially with others who otherwise live quite far, and sometimes they have neighbors that they actively avoid, uh, that's pretty useful, because it gives us information on how much your microbiome similarity might be affected by your exposure to each other, like the kisses, like actual social interaction, versus your exposure to the same space, where you might be getting the same microbes from the soil, from the diet, or just from each other indirectly through the environment. And we can also look at things like habitat similarity and stuff to control for other things that might affect microbiome similarity than the actual social contact. And then build this kind of model where we predict similarity, a proportion of shared taxa with habitat similarity, home range overlap, social association, and a bunch of other covariates like sex and age similarity. But you notice that all of these variables are of a pair. So you're looking at pairwise regression. So each data point in this data set is a pair of individuals. So it's obviously those pairwise comparisons are not independent. So how do we use a linear model to predict that? And that's where we use Bayesian, something called Bayesian multi-membership beta regression. I use it in BRMS package in R. It's a GLMM, like a generalized linear mixed model. Just like many of the ones that biologists and many data scientists use as a workhorse, but instead of having kind of random factors that describe higher level categories for each data point, it has something called a multi-membership random effect, which gives each data point membership in multiple higher categories. So it's basically I put this picture here. I use this to explain to my biology students how this works. So this may be not so much for like kind of this kind of audience, but basically normally when we model things like individuals, we're interested in variation of individuals, like size or color of these mice. And then we're interested in like hidden autocorrelation between them, like spatial autocorrelation, genetic autocorrelation, and we want to kind of explain that away so we get to these patterns between individuals. But this is, we just flip this whole picture upside down. So suddenly we're interested to modeling the pairwise values between these individuals, such as their distance from each other, their social association to each other, their similarity of their microbiome. And then we use the identities of the individuals as the random structure in the model. So we give basically each so in this model, for example, each data point belongs to two higher categories, which are the individuals that connect that pairwise connection. So for example, my social association with Tuomas and Tuomas's social association with Jeba belong both to the same higher category called Tuomas. And then they also belong to these two other categories, which are the other ends of these lines connecting. Yeah, and then also I've been kind of nerding out, like actual microbiology information, looking at things about the bacterial genera themselves, categorize them for aerotolerance and sporulation ability, because I wanted to know whether the ability of the bacteria to tolerate oxygen and persist in the environment might be somehow related to how they transmit through social or spatial routes. Okay, so some results. Is there an effect of social networks on microbiota? I can pretty much say yes, because I now repeated this in 4 different data sets. Here's 2, which ones I described. And in both of these, you can see that there is an effect of, there's independent effects of habitat similarity, spatial overlap, and social association, or here is spatial proximity and social association, on microbiome similarity. But the social association effect is manifold stronger. So you, by having actual social associations, you share much more microbiome than by just being exposed to the same space. We've also controlled this against genetic relatedness and diet similarity before, so, and those did not have any effect in this system. So yeah, there's a strong signal, and actually, I wish I had the third plot here from the third data set, so you could see how we see the same pattern in different data sets, but as we go ahead, like more and more later in time, like from 2013 to 19 to 22, the credible intervals shrink and shrink and shrink. And that is just because I become better at lab work. Like when I'm a master's student, I fuck up everything, sort of contamination, lots of uncertainty in the data. Then here, this is the above one is my PhD work. Still quite a lot of wobble, but very clear effects. This is me as a postdoc, totally like nailed it. So just an example of how much it actually means, how well the data is prepared. So what kind of microbes are spread through what kind of contact? So then I repeated this model by just looking at where the response variable would be just the proportion of shared aerotolerant or anaerobic microbes. So those who can tolerate oxygen or can't tolerate oxygen. And we find pretty clear signal that when we're looking at oxygen tolerating microbes, we lose the social signal. And the spatial signal gets actually stronger. And kind of the opposite happens when we look at anaerobic microbes, which is quite clear evidence that the social signal is driven by anaerobes and the spatial signal is driven by aerobes. Makes sense, right? Because you need to tolerate oxygen to spread through space, and apparently you also need the anaerobes really need the social contact to spread. And to do this a little bit more, even more in detail, I wanted to cut this data down into individual bacterial genera and calculate for each bacterial genera some measure of transmissibility through social or spatial routes. And the way I did this was that I used this kind of leave one out approach where I dropped each bacterial genera out of the data in turn, then rerun the model and looked at the spatial and the social effects and see what happens to the uncertainty around those effects when we leave one genus out. And with the idea here being that if you have a very influential, very socially transmissible taxon, if you drop that out, then the social effect should get wider credible intervals. And then if that taxon was just introducing noise, then it should get actually more credible. And then we use this, the relative increase or decrease in credible interval width per genus dropped with some other corrections. to calculate basically each gene is like an importance for social signal, importance for spatial signal, and importance for habitat similarity signal. But I'm not talking about that now. But and then I compared that to what I knew about the actual biology of these bacteria, and we found out that the social transmissibility or social importance is highest on the microbes that are anaerobic and non-spore forming. So those who really cannot in any way spread through the space. And then conversely, aerotolerant spore formers have the highest for the spatial importance. So it also makes sense in light of the earlier evidence. But this is, I think, a nice way of, like when we look at this whole community level sharing in networks, how do we then kind of zoom in and disentangle, so which parts of the community are driving which signals, so we can do this kind of live on out approach, for example. And also, importantly, these socially very transmissible microbes tend to be the very healthy gut microbes, because these anaerobic non-spore formers are the ones that really, really matter for your health. Okay, maybe I still have a couple of minutes, so I can show you the last bit of this. So what are the most diversifying positions within a transmission network? So this is like kind of my most favorite question. So all of this previous stuff has been just me establishing how these spreading processes happen, because I wanted to ask this question. I wanted to know that when we have social networks, where should you be in the social network to get the most, most diverse microbiome? And that's the same question we could ask in information networks, like what kind of positions are more diversifying for your information, or where do you end up having very poor, like kind of you get the same news from everybody, you know? And that's very important, because just like in information networks, in microbiomes, diversity is really good. So diverse microbiomes make you healthier, and when you lose diversity, that can like in many ways render you kind of sick. So so we want a diverse microbiome. So how do we optimize that? But then also, how do we do that in a way where we avoid pathogen transmission? Like how do we get the good stuff but not the bad stuff? That's like the core of my research. And I started doing this by looking at now not using a pairwise model anymore, but like for every single individual mouse, now calculating something like a position in their social network and a density of others around their spatial centroid location. And yeah, this was done by using a this density I calculated by just looking at the inverse of scale distance between location centroids of individuals. So it's a very simplistic method, but actually works pretty well compared to some more fancy methods. And the social network, I just use these existing so-called centrality metrics that describe someone's position in a network. So for example, degree is the number of contacts you have, and then eigenvector centrality or transitivity describe how much you're in the middle of a cluster, so how much your friends are friends with each other. And then conversely, bridgey metrics like bridge propensity and betweenness on the here maybe better shown, describe how much you connect otherwise disconnected parts of the network, so how much your friends are not friends with each other. And then I use these metrics to predict microbiome richness, so how many different microbial taxa does an individual have, with some covariates. And what I find, first of all, is that this bridge type social centrality is more related to microbiome richness. So here you can, if just look at these networks, you see that when you compare the microbiome richness as a node size, it looks more like the one where the betweenness is the node size than the eigenvector centrality. This is just a visualization of the pattern. But in the model, we actually find that it's these bridgey metrics that significantly positively predict, like have an effect on microbiome richness, whereas, for example, spatial density actually has a significantly negative effect, which is surprising. But I think it's because of stress. It's stressful to be sharing resources with many others, and that can lower your microbiome richness. But these clustery metrics don't have any effect. And I think that's cool, because that's what I expected. Well, when you're connected to, it's not just the number of friends, it's how much your friends are from very different social circles that is enriching, that can lead to you having different stuff from different individuals. When you're in the middle of a cluster, like all your friends are friends with each other, kind of a hipster king queen situation, then you actually get the same microbes from everybody, because they already shared them with each other. And it's a bit same with information networks, you know, like when you're in the middle of a scene, it's a little bit impoverished position in terms of information flow because everybody has the same opinions already and the same information sources. Whereas when you have one friend who's a left wing or one friend who's right wing, then that's when you have very diverse political opinions. It's the same process, basically. And the next steps here, which we already started doing, but I don't think I have time to discuss these results. But basically, in addition to diversity, one other aspect that's really important for your health is your microbiome stability. And since we have this long time series where we have like up to 30 samples per individual mouse in a short time period, we can use those to calculate each microbiome stability, like how much it changes over time, and then use those same centrality metrics to predict the stability to see which positions in the transmission network accumulate the most stable or unstable microbiome. And here I am working with Ville and Leo to develop not just so we can, what we are already done is just calculate basic turnover rate, so how much microbiome changes over time, but it also matters how much it changes stochastically or deterministically, because in an ecological system like microbiome, deterministic changes that are driven by some, like for example our immune system, can be very adaptive. It can be, for example, you adapt into a new diet or new cold season. But stochastic changes, when things completely go out of balance and change chaotically, those are usually a sign of illness or sickness. They are like your microbiome is not doing well. So that's especially interesting when we want to see which parts of this transmission network tend to have those kind of destabilizing positions. But we don't know that yet. Just comparing, saying that pathogens actually, both viruses like COVID, but also in our data, parasitic worms, tend to accumulate in those clustery positions. So that's been established in many different systems. So I think it's interesting to show that these healthy microbes accumulate in these bridgy positions in network, whereas pathogens might be accumulating in these more clustered positions. So if they actually accumulate in different parts of the network, then we can start asking questions like what kind of networks theoretically would maximize the spread of good stuff but minimize the spread of bad stuff. That is all I have to say today. Thank you. There's one there.
How do you assign poop to a specific mouse? And can
Good
you
question.
control for the age of the poop?
Oh yeah, very good questions. This is the kind of real biology questions. So when we trap the mice, they go into this little live trap, like a little box. There's peanuts there and some nice cotton wool. It's like a hotel, and the door closes behind, and the mouse is trapped there for like a couple of hours. And then we go and we tag the mice, or we read the tag if it already has a tag, so we know which mice was in the box, and then we can collect the poo from the box, and then we can use the how long time the mouse, like how since we put the trap on as a covariate in the models, like how old the poo is. But of course, it's a bit of a mix because it'll be like some of the pellets we collect are older than others, and I do know that actually has an effect. So that's some variation we are not currently like fully controlling. So I've noticed that if we set the traps in the night and then collect them in the morning, or if we set them in the night and collect them in the night, that does actually make a difference in what we find. But yeah, I hope I'll find a better kind of workflow in the future to account for that.
Here a question, Ville Pimna from Oulu University. Very nice work on, but could you say a little bit on the factor that when you look on the different taxonomic depth, how that affects on the signal?
Yeah,
because on my interest is in the oral and vaginal microbiome in humans, and I think that's in the contact networks is very interesting.
Yeah, so a lot of this other kind of spread of microbes in network stuff has been done on strain level, and that's because people have wanted to actually identify the exact strains that come from someone and go to someone, like mother to infant, for example. But I use a very different kind of method, because first of all, even when we look at this ASV level data, we find this very strong signal, so it hasn't been like very useful to go any deeper than that. but and I specifically for the importance or the kind of transmissibility signals, I used genus level, because these phenotypes, these bacterial traits that we're looking at, like aerotolerance and sporulation ability, are usually quite conserved within a genus. So it doesn't make any difference if we look at it on the species level or strain level, because all microbes in this genus will be aerotolerant or anaerobic anyway. So that's kind of the level where the biological variation is. So that's also something I wanted to say, that a lot of these bioinformatics developments is often kind of assuming that higher taxonomic resolution is always better. But I think when you're looking at these kind of whole community signals, like when you want to not target one specific thing, but like look at how whole community spread, actually lower taxonomic resolution is better, because it gives you you have to be on the scale of where the biology happens, like what's the meaningful kind of level.
And when you look on the signal between communities, for instance, with mice,
Yeah.
do you actually see that there's a isolation by distance on certain at genus level, or if you go deeper on species level as well, or the signal is actually very similar on?
So actually the pairwise model I showed, I haven't run that in different taxonomic levels. So that's based on ASV level. I haven't checked how it would be in genus level or like kind of more coarse levels, but that's a good idea actually. I should probably see. I think it will degrade pretty fast. Like I think genus you might still see it, but I think not above that, because if I look at where the variation between the individuals are, it's highest on the ASV level. So, and then genus level, they still have some variation, but on family level, they're all quite similar. There's not so much variation anymore.
For the sake of time, we don't have time for more questions, but just one last one here online. Could you apply this method to other species, and what do you think would be another interesting species you look at?
Oh yeah, so actually, so the mice are here just a model species. This shouldn't be a mouse-specific process in any way. You could definitely apply to this anything, and not like I'm trying to make these methods and these insights as general as possible. I know it sounds a bit specific to be studying wild mice, but I think there's no reason to expect why this would be the case in wild mice, but not in humans or not in some other individuals. So it's just about linking contact networks with similarity of microbiomes, and I would like to do this on humans. It's just pretty hard to get this kind of data from humans, and the most importantly, what's the hardest part is that for these kind of models to work, we need pretty complete social networks. We don't need, the more you have missing edges, especially for the centrality kind of analysis, the missing edge data is creating exponential amount of bias and noise. So if you find any data sets where you have actually complete social networks of humans where no one is missing, then, or like even 80% or something, then I'm really happy to hear.
All right, thank you very much. So let's all thank Aura.
Thank you.
And now I'll give the word to Mike, who's going to chair the next session.
Great, thank you. So we have 4 talks. Awesome. We have 4 talks. I'll do the same thing as before, so I'll